Hello, my name is Mohamad Fazel, and I work as a postdoc at the Presse lab, Physics Department, Arizona State University. In our lab, we employ cutting-edge machine learning techniques to develop tools for fluorescent microscopy data. Today, I'm going to talk about one of our recent published papers on fluorescent lifetime imaging in collaboration with the labs of Professor Digman and Professor Gratton, with the title High Resolution Fluorescent Lifetime Maps from Minimal Photon Counts. Here, I will explain fluorescent lifetime imaging microscopy, otherwise known as FLIM. In FLIM, samples are labeled with fluorescent molecules. In this slide, we see a simple diagram of the quantum structure of energy states of such fluorescent molecules. These molecules are often in the ground state, but can absorb a photon from the excitation laser and go to the excited state. They spend a time tau in the excited state and then emit a photon and return to the ground state. Different fluorophore species have different lifetimes that can be used to distinguish them. For example, here we have a sample whose different parts are labeled with different fluorophore species, red and blue species with different excited state lifetimes. Such samples are often scanned using a laser scanning confocal microscope, where each spot of the sample is illuminated with a certain number of laser pulses. Here, the pulses are shown by pink spikes. A few of the pulses lead to a photon detection, shown by curly arrows. The photons collected from different spots of the sample are arranged into a 2D pixel array representing the raw film data, as you can see here. This raw film data can be processed using different existing film analysis methods to obtain the lifetime and the distribution of the corresponding fluorophore species, termed lifetime maps. Such film techniques have multiple applications in biological sciences, including mapping out the variations in temperature, refractive index, pressure, molecular concentration, etc., within subcellular environments. For instance, variations in the concentrations of free and protonated NAD can indicate changes in metabolism due to disease such as cancer. The analysis of raw film data using the current techniques faces multiple challenges, including 1. Learning the number of lifetime components present within a given dataset. 2. Dealing with a broad range of lifetimes, including lifetimes longer than the interpulse window and closed lifetimes. 3. Providing lifetime maps with resolutions better than the data pixel size. 4. Taking full advantage of all the existing data, such as information carried by empty pulses. And 5. Reporting uncertainty over the estimated parameters. We have previously addressed the first challenge for only one pixel. Here, we tackled the rest of the challenges for wide field film. As we discussed in previous slides, each pixel of the data contains photons collected from a specific spot illuminated by a train of laser pulses. Here, the data are 1. If a pulse leads to detection of a photon or not, meaning empty or non-empty pulses, and 2. Photon arrival times. Data from these pixels can be used to construct the likelihood, which is the probability of the data given the parameters. These parameters include lifetimes and the corresponding lifetime maps. However, within the Bayesian framework, we work with the posterior, which is proportional to the likelihood times priors over the unknown parameters. Since here we are learning a map with correlated values, we assume a Gaussian process prior for lifetime maps, which is simply a multivariate normal distribution. For lifetimes, we take gamma priors on lifetimes to guarantee positive values. Here, we won't derive the exact form of the posterior, but it has a complicated and non-standard form. This slide shows results for a lifetime map using Gaussian processes, the raw film data is shown on the left. On the right, we see the found map by our method. Here on the right, we see a mix of simulated ground truth maps for 2 lifetime species over a 6 by 6 micrometer square region, and on the left, the lifetime maps deduced by our method. As we see, they match pretty well. We simulated data with the same lifetime maps shown in the previous slide, but lifetimes that are only 0.5 nanosecond apart. This would be a challenging case for the current film data analysis methods, but as we see in the histogram, our algorithm distinguishes and learns these lifetimes. We also use the same lifetime maps to simulate data with a lifetime which is 2 times larger than the interpulse time, which is 12.8 nanosecond here. As we see, our algorithm still distinguishes and learns the 25 nanosecond lifetime, although the uncertainty is large, indicating a wide posterior for large lifetimes. Finally, in this slide, we show results for experimental data. Here we see the ground truth maps for lysosomes and mitochondria in green and red, respectively. Yellow is the raw film data from the mixture of these two maps. On the right, we see the lifetime maps learned by our algorithm, which match with the ground truth maps with less than 5% errors on average. Thank you for watching my video. Here, I want to acknowledge our collaborators, Professor Digman, Professor Gratton, Dr. Sipiani, and Dr. Walmit-Jana. I hope you learn a lot about what we do to map fluorescent maps. Thanks.